Hello everybody, welcome back. Today we're going to be talking about Chapter 8, the Introduction to Carbohydrates. This isn't a super long chapter, but it's a really good thing, good chapter to sort of set up a lot of information. So let's just jump right in. So looking at our objectives, first off, we're going to describe the distinguishing categories of carbohydrates, some of the different isomers, epimers, enantiomers, and optical rotation. Lots of big words there. List the functions of carbohydrates in biological systems. Classify monosaccharides. We've talked about these before, and we've mentioned that they will continue to pop back up in a whole bunch of classes. So classify the monosaccharide sugars and give examples of the important monosaccharides in biological systems. Describe O-glycosidic and N-glycosidic bonds and where they are found. Pay attention to the O and the N. That'll give us a clue later on. Describe the structure of disaccharides and polysaccharides. So if we know what a monosaccharide is, hopefully we remember that a disaccharide means two sugars together, poly meaning greater than two. Occasionally we'll say trisaccharides and polysaccharides are, trisaccharides are 3 and polysaccharides are 4 or more, but you can also call polysaccharides anything larger than a disaccharide. Be able to identify various modifications of sugars and explain the purpose of these modifications. When we talk about modifications, we're talking about things that get attached. Think like phosphates, not methane, methionine, there we go, get it methylated, those sort of things. And finally, give examples of glycoconjugates, their structures, and their roles in mammals. So introduction to the carbohydrates. They are an essential component of all living organisms. All living organisms, we all need carbohydrates to survive. They are also the most abundant class of biomolecules. Name carbohydrates, polyhydroxy compounds. Polyhydroxy compounds. That's an interesting looking word. Let's take a look at that real quick. Polyhydroxy compounds such as cyclitols, acetyl polyols, and sugars are produced by a wide variety of organisms under stressful conditions. Let's look at polyhydroxyl. Define. Containing more than one hydroxyl group in the molecule. Okay, so polyhydroxyl compounds, so sugars will contain more than one hydroxyl group. They contain carbon, hydrogen, and oxygen. Their role, they have both a structural and a metabolic role. Metabolic referring mostly to energy when you hear the term metabolic. Now glucose, we'll talk a lot about glucose. Glucose is the most important carbohydrate. It is the major fuel of the body, and understand that without glucose, we really cannot survive, okay? Either we are ingesting something that can easily be broken down into glucose. So think of any foods that we have, the macromolecules, any food that has carbohydrates in it, or our body is going to produce glucose by breaking down our non-glucose sources, so our fats and proteins. And if you remember, that is called gluconeogenesis. Okay? Making of glucose from non-carbohydrate sources. So can make all other carbohydrates in our systems. This is why glucose is so important. Glucose is like that essential building block for all of this stuff. Glycogen for energy storage, ribose for nucleic acid synthesis. This is DNA and RNA. So DNA is needed in every single one of our cells. So in order to replicate our cells, we're going to need ribose, which can be made from glucose. And RNA is designed for copying, essentially for copying our DNA in order to make actual proteins and genes and make our DNA actually functional. We'll talk more about that. I'm not sure if in this class or if in another class where we'll go deep into the formation of DNA and kind of DNA and RNA and how that whole process works. Okay, molecular and cellular recognition, adhesion, lubrication of joints, signaling molecules, structural and protecting elements in connective tissues. So glucose can be used to make all these other carbohydrates. All of these are jobs of carbohydrates. Classification of carbohydrates. So this is actually going to correct something I said a little bit earlier. So we want to, if you took a note, let's correct it down here, correct it back up here. So we have aldehyde or ketone derivatives of polyhydric alcohols are one type of carbohydrate. Monosaccharides, basic unit of carbohydrates. So this is one single sugar. Disaccharides contain 2 covalently linked monosaccharides. Oligosaccharides contain 3 to 6 monosaccharide units. And polysaccharides, we will go with this definition that they're greater than 7 monosaccharide units. So our polysaccharides are quite large. Our monosaccharides, aldehyde or ketone derivative of straight chain polyhydroxy alcohols having minimum of 3 carbon atoms. Okay, so this is a little bit confusing, so let's break this down. So our aldehydes are our ketones. Aldehydes and ketones are kind of their own special little thing that we'll talk about, okay? It's a derivative of straight chain polyhydroxy alcohols. So polyhydroxy, we saw that word before. Polyhydroxy meaning it has multiple hydroxyl groups. And then alcohols, those alcohols are going to have that specific combination. Let's see if we can get a good polyhydroxy alcohols. Compounds that possesses more than one alcohol group are called polyhydroxy alcohols. So the hydroxy, that OH group, having that OH group is actually what is going to be making it an alcohol. The functional group of an alcohol is the hydroxyl group. Okay, but these are derivatives of the straight chain polyhydroxy alcohols having at least 3 carbon atoms. Okay, here's a thing that we'll want to remember, just name-wise: aldehydes are aldoses and ketones are ketoses. When we get into talking about the condition ketosis, that's what we're talking about. When our body, we have all these ketones. So these aren't going to be our primary sources of sugars, of carbohydrates, but they will be used when necessary, when needed, okay? So the general formula for these are CH2O, N, that means to the nth, so like multiplied by N. So if we think we can have CH2O, C2H4O2, etc., etc., etc. And here is some charts. This chapter is going to have a lot of kind of memorization, to be honest. This is something that I would recommend just kind of drawing these charts out over and over and over. Or even if you can remember like TTPH, GERG, DERF, remember like the first letter, and just remembering like the small differences. So these ones are oses. Just usually it'll be ros, bos, cos. These just kind of trying to find different ways that you can remember how to differentiate these, because the kind of questions that you're going to see on this portion of the material is really going to be as simple as which of the following represents an aldose with 4 carbons, which of the following represents a ketose with 6 carbons, that sort of stuff. If I do recall as well, in Dr. Sarkar's actual lecture, he does give an idea of which ones of these we should really focus on and remember, if I remember correctly. I don't know if it's in this specific PowerPoint or if he has another one where he talks about it, but he will go over. Here we go. We can actually see in a lot of these. So see how these ones that he has boxed in. These are these ones that are boxed in. Focus on these guys. Knowing the names of those ones and like how many carbons they have will help you a lot in answering questions for this exam, this material. Okay. Structure of some monosaccharides. So we have glyceraldehyde. These are all just a bunch of these different examples and showing kind of those functional groups on them. And see these OH, 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 all of these. That's what makes it that polyhydroxyl alcohol. Okay, stereoisomers. Remember when we talked about isomers? An isomer means that it has the exact same chemical formula, just that it's kind of rearranged. It's arranged in a different way. Or actually, so the stereoisomers, talking about, it's like a reflection, that it's reflected over. So it's the same structure, but just the bonds are on opposite sides. So compounds that have the same structural formula but differ in their spatial arrangement are known as stereoisomers. The presence of asymmetric C atoms allows the formation of isomers. The number of possible isomers of a compound depends on the number of asymmetric carbon atoms and is equal to 2 to the nth power. So glucose has 4 asymmetric carbon atoms. So that means it is 2 to the 4th power. Hence, or so therefore, glucose has 16 isomers. From this portion, this equation, understand what this equation is really saying. What this is saying is that when the number of asymmetric carbon atoms, so let's actually pull up a glucose. Here we have our glucose. So see each of these here, how they're slightly different. OH, OH, OH. Hydrogen on one side and OH on the other side. What this means is that, so it's actually going to be one, if I remember correctly, it is one, I think 1, 2, 3, 4, these 4, if I remember correctly. These 4 essentially can have different arrangements. So these are the asymmetric ones, where they have different things on each side, right? The OH and the O's, or the OH and the H. So any of position of these, if the hydrogen was on the opposite side, or the oxygen and the oxidizing, whenever these are switched, right? Every time one of these switches, it becomes another isomer. So this one switches, isomer. This one switches, this one switches, this one switches, and so on. And that's what we're talking about. This is what it is: 1, 2, 3, 4 asymmetric carbons. And those 4 asymmetric carbons are what allows it to have all of those different isomers. Or there's just the same structure, just slightly different arrangement. And so that equation, that 2 to the nth power, is how you solve for how many that there are. This will be some sort of test question talking about the possible isomers, and it will either be 2 to the nth, or he'll ask you specifically about glucose being 2 to the 4th. Or he'll say like this, there's this sugar, and it has, let's say, 5 asymmetric carbon atoms. How many possible isomers does it have? And so then you need to do that calculation. Okay, D isomer versus L isomer of glyceraldehyde. So you have D isomers where the OH is on the right of the chiral carbon, and L isomers where the OH is on the left of the chiral carbon. See, just that's flip right there. Doesn't focus a lot on this. Kind of just one of those things that you kind of throw in. Boards every now and then will throw questions like this in there, but not super common, so I wouldn't worry too much about a lot of these isomer type stuff. Optical isomers. So the presence of asymmetric carbon atoms also confers optical activity on the compounds. When we're talking about optical isomers or optics, this is a process where we sort of look. We actually have a machine that uses light that go through the solution to check and see kind of what isomers that we have. So the presence of asymmetric carbon atom also confers optical activity on the compounds. So a beam of polarized light passed through a solution of optical isomers rotates the light either to the right, D, dextrorotatory. You'll learn this about dextro. Remember this prefix, dextro. Dextro pops up a lot later on when you're talking about stuff like scoliosis and some other anatomical terms. It just means to the right. Or the left. Left is levo. Same thing, remember that, levo. Remember levo. Levorotatory or negative. Accordingly, a compound can be designated as a D+, a D-, an L+, or an L-. Again, no kind of in general what an optical isomer is, but other than that, like we won't get too deep into all of this stuff. So all monosaccharides are asymmetric except for dihydroxyacetone. Whenever there's an exception, we like to ask questions about the exception. So remember that word. Optically active isomeric forms are known as enantiomers. Non-superimposable mirror image. Non-superimposable mirror image. Know the definition of this. Definitions come in handy, and they like to ask questions basically strict off to the definition. Let me see real quick. He doesn't spend a ton of time talking about these. All of the monosaccharides are asymmetric. Optically active isomers. Yeah, this is literally about as deep as we go in all of this. Just like know the definitions of kind of stuff. Know what an enantiomer is, and you should be okay decently with that. Here we go. Only D isomers are used to synthesize carbohydrates in humans. Only D isomers used to synthesize carbohydrates in humans. D isomers. Okay, so real quick. When we get to an exam and we're looking at the different questions and the different answer choices, 99% of the time, if we see an L isomer, so L-aldolase, aldolase, for example, L-glyceraldehyde, L-erythrose, anything like that, we can ignore it. Okay? Because we only want the D isomers. So if it has an L, cross it out and say, no, 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 that is not the answer. All of these here that are boxed, these are the ones we want to know. Make yourself, I would highly recommend making yourself a couple of little charts to go over these. Okay? Just like this chart that was up here. Mark off which ones, mark the ones that are down below that are squared, and add more. So just to keep them straight, where there's 3 or 4, 5 carbon, 6 carbon, okay? Really, really know these ones. I would say if you know the ones that are squared, I remember when I took the class, I think he threw in one question about one of the ones that wasn't squared, but all the other questions were about ones that were specifically squared. And there's a fair amount of these that are squared. So I would highly recommend also making some kind of mnemonic or some way to help remember these. Whether that be, like we talked about, DERXF, so Dirk's F. If you can remember something, so like ketone. So ketones, di-earth, something, anything that you can help remember. Di-earth, X-ribs, finally. I don't know, something you can, there's also like mnemonic generators that you can go and you can put things in, but anything that you can remember, you can do to help yourself remember these, do that. It takes a couple times. There's some research that says you need to see something about 17 times in different formats in order to fully retain it. So I would, even flashcards, these would be great for flashcards, just to remember how many, whether are they a ketone or an aldehyde, how many carbons that they have, and if you can remember those couple of things, the test will be so much easier with any question that you can throw at you with these. Epimers. So they are isomers differing as a result of variation in configuration of the OH and the H groups on C atoms 2, 3, and or 4 of glucose. So we are specifically talking about glucose right now, okay? We have our glucose. Mannose is an epimer at C2. Galactose is an epimer at C4. So glucose is the big one that we want to remember. And actually these ones, so we'll see mannose and galactose count as other monosaccharides as well. The monosaccharides that we'll want to pay attention to, glucose, galactose, let me quadruple check. Fructose. Which other ones do we get tested on? Mostly these three, glucose, galactose, and fructose. But you will also see occasionally pop in there ribose, xylose, and mannose. Okay? These three for sure focus on. But every now and then ribose we will see pop in there. Mannose and xylose is almost never, but those top three are the main ones that we'll focus on for any and all classes. Okay, glucose, the dominant monosaccharide, 3 structural representation of glucose. So this is how glucose actually. So these are just some different ways of how their chemical formula, the chemicals are represented. Where in reality, like this is how it's written out more in like a straight chain, just to show its whatchamacallit, the like its formula. This is more of its actual shape bent down a little bit, and there's even more like realistic, I guess you could call it. 2 cyclic forms of D-glucose. So we see just these 2 different alpha versus beta, the slight difference. Again, we don't focus a whole lot on all of this, okay? If anything, I would just make a note that the C1 carbon in glucose is the one that is the difference between the alpha and the beta. D-fructose, same thing. Don't spend all of your time memorizing these shapes and stuff. Don't worry about that. Just know like how many carbons there are and know what fructose is, okay? Ribose and deoxyribose sugars are used for nucleic acid synthesis. This is actually why ribose, if we talk about RNA, this is a ribose nucleic acid, and then DNA is a deoxyribose nucleic acid. So what they'll be made up of, DNA is the deoxyribose, RNA is the ribose. Look, deoxy, it's missing an oxygen right here. That's the difference between ribose and deoxyribose. There's 1 oxygen difference. Sugars form glycosides with other compounds and with each other. When we talk about glycosides, we're essentially talking about just disaccharides, where they're going to combine together through a hydrolysis or a condensation reaction. Again, don't get all caught up in all of this stuff. Just understanding the concept that they're going to combine. Our glucose will combine with glucose to create a maltose. Okay. Some other common disaccharides. These are the big ones you want to know. No lactose and no sucrose. He will have questions, so like. So for example, lactose, you want to know the monosaccharides that make it up. It's a galactose plus glucose. Sucrose is fructose plus a glucose. And you can just, it is what this is saying down here. It's got like the proper quote unquote naming for it. But just know this, galactose plus glucose becomes lactose. Fructose plus glucose is sucrose. And maltose is glucose plus glucose. Polysaccharides. Homopolysaccharides, homo meaning same, contains same monosaccharide units, so it contains the same monosaccharide, so that would be like glucose and glucose, galactose and galactose, fructose and fructose. If you add the same thing together, it is a homopolysaccharide. Heteropolysaccharides contain different monosaccharide units joined to form a linear chain as well as a branched chain. So glycogen, for example, is a polysaccharide. This is the animal storage form of glucose. And you also have starches, which is the plant storage form of glucose. Keep that clear that glycogen is in animals, starches are in plants. And these main 2, amylose and amylopectin, these will come back a bit in 5, I believe, in, let me double check. Maybe it was try 4. Yeah, in try 4 with clinical nutrition, these guys will pop back up. So keep an eye on these, because we'll talk quite a bit about amylose and amylopectin in that try. These are just more pictures. Glycogen. This is the main one that we're going to focus on right here. This right here, these two phrases right here, is exactly how he's going to ask a question. If he asks a question about these, this is exactly how he's going to ask it on the test. So look at those definitions. It's a linear polymer of glucose versus a branched polymer of glucose. Amylose is linear, amylopectin is branched. Other polysaccharides, cellulose and chitin. We'll get more into both of these later on. Just look at the names. You might want to look at the linkage as well. I'd make a chart if I were you. Make a chart of the names and then the linkages as well. Because every now and then he'll throw a couple of questions of the linkage. And that, unfortunately, that's just straight memorization. It's not anything exciting. More images, lots and lots of pictures. Glycosaminoglycans. These are components of the extracellular matrix. So these are going to be part of when we're way up at the top. When we talked about some of the functions of these sugars, right? We have structural and protecting elements in connective tissues. That's what we're talking about with our glycosaminoglycans. The examples: hyaluronic acid and chondroitin sulfates. Hyaluronic acid, this is a big one. You'll hear a lot about hyaluronic acid in a couple different classes. So again, with this, just focus on the definitions that a glycosaminoglycan, a component of the extracellular matrix. The function of that is connecting tissue. Hyaluronic acid is one of those big connecting tissues. Chondro, this base chondro means cartilage. So this is going to have to do with cartilage. Okay. If I'm not mistaken, hyaluronic acid is something that, so hyaluronic acid, a natural substance found in the fluids in the eyes and joints, acts as a cushion and lubricant in the joints and other tissues. This hyaluronic acid will come up a couple of times as well in other classes. Exactly which ones are slipping my mind right now, but that is one. Keep an eye on that one, because that one will come back quite a bit. Glycoconjugates, proteoglycans, proteos, protein, glycans. So it's protein mixed with our sugars, glycolipids, and glycoproteins, just depending different forms of it. I would go ahead and pause right here and real quick do a Google search and define each of these terms so that you get a better idea of exactly what to expect with each of those. Maybe a couple of examples or kind of their general function to help you remember those. Oligosaccharide linkages in glycoproteins. So sugars are attached by O-glycosidic linkage or N-glycosidic linkage. So our O-linked, there's the O, and our N-linked right there. So it just depends on what part, kind of the formation and where they're going to end up binding. Literally O, oxygen, N, nitrogen, how they're linked. And that's the whole chapter. Because this will get into the next stuff. So a lot of this chapter, to be honest, is kind of filler material, in my opinion. There will be a handful of questions that get pulled from this material, so you want to look over it. But main focus on definitions, like oligosaccharide, oligosaccharide linkages. Like, take some time to go through this chapter, look at definitions to prepare you for what's coming down the road, okay? Because the glycoconjugates, the glycosaminoglycans, hyaluronic acid, chondroitin sulfate, this terminology, and a lot of these sugars, the amylose, amylopectin, cellulose, chitin, etc., all of this stuff is going to come back. So take the second now to look it up, so that way you'll be good to go and you'll understand, at least have a base knowledge of what it is when we start to actually take those things, that terminology, and put it in and plug it into other things to apply it. Okay. Well, that's the end of this chapter. So hopefully good luck studying, and I wish you all the best of luck.